Cell morphology captures a huge amount of biological information like differentiation state, stress response, or Bragg effect. Here I have this public dataset of human pluripotent stem cells captured in different states of differentiation, and I will show you how to use morphological gating in Cytely to isolate and compare distinct cell populations. First, I've created a workflow to segment the cells. These cells were stained for both the cytoplasm and the nuclei, so I segmented the nuclei using segment cell nuclei, and I used them as seeds to create a mask for the cells from the cytoplasm. You can use this drop-down menu to select other images to make sure that the segmentation mask is accurate for all cell types. The cells were also stained with mitotracer for mitochondria, so I connected this channel along with the cytoplasm and the nuclei channel to a DICT node. By connecting the DICT node to cell morphology, we can measure the signal intensity from all three channels within the masks. Here you can see the results and create different plots and charts to explore the subpopulations in your sample. Down here you have an overview map where you can see all the cells that were detected. Here I plotted the area versus the perimeter of the cells. You can see that there is a subpopulation of cells that are much larger than the others, and these should be the astrocytes. We can create a gate to isolate and inspect this subpopulation, and the cells will show up down here. You see that when we create a gate, the population becomes isolated in the following plots and charts left to right. You can also inspect one cell group at a time by deselecting the others. Now you can see that we can distinguish two subpopulations in this group, which could be due to the cells being in different cell cycle stages, for example. Here I plotted the mean intensity of the mitochondria against the mean intensity of the nuclei, and again we can create a gate to isolate any subpopulation that could be of interest, for example this one. You can also click on a single detection, and by selecting Show in Map, you can see where it is placed in your images.